relevance, right, have some importance to differences in the way genes are expressed in bacteria versus eukaryotes like us. Different DNA? Do we have different DNA? We have 46 chromosomes, and we have other parts to our DNA that are different, but the molecule of DNA itself is the same no matter where you look, yeah? So there are some differences. What else besides some differences in architecture of DNA, if not structure of DNA? Yeah. We've got a nucleus and they don't, yeah? Does that matter? Yeah, in order for a messenger RNA to get to a ribosome, what does it have to go through? The nuclear membrane, yeah? There's a membrane around that nucleus, yeah? And so transcription, right, turning DNA into messenger RNA happens where in a eukaryotic cell? In the nucleus, yeah? What about in a prokaryotic cell? Where does it happen there? In the cytoplasm, yeah? Just in the plain old fluid of the cell, yeah? What about translation? It happens in eukaryotic cells in what structure? Yeah, on the endoplasmic reticulum on a ribosome, yeah? Right? And in a prokaryotic cell? On a ribosome, not necessarily on endoplasmic reticulum, or definitely not on endoplasmic reticulum. They don't have it, yeah? So, but on both it's happening on a ribosome. The ribosome is in the cytoplasm in a eukaryotic cell and in a prokaryotic cell, yeah? You agree? Right? So the difference is that eukaryotes have a nucleus, and therefore transcription and translation are separated by the nuclear membrane. Yeah? Does that make sense? Whereas in a prokaryote, they're not. They happen in the same liquid space. Yeah? So eukaryotes have a nucleus. Most of the differences between prokaryotic and eukaryotic central dogma come from having a nucleus. Okay, so there's a couple other wrinkles here, but let's talk about it. So, eukaryotic transcription and translation must happen separately, right? We separate them out physically by the nuclear membrane, right? We do transcription inside the nucleus. We do translation out in the cytoplasm on the ribosome, yeah? Messenger RNAs leave through a nuclear pore, a little hole in the nucleus, the nuclear membrane. Yeah? What do you think? That's a great question. We also have slight differences in our ribosomal structure. As it turns out, the nuts and bolts are pretty similar. Yeah? Similar enough for me not to talk about the differences. Yeah? There are definitely differences, but they are not anything I'm discussing. Here is an important difference, though, is that eukaryotes have DNA architecture, as somebody mentioned, a little different. We have linear chromosomes, not circular chromosomes. Humans have 46 and not 1. Yeah? But furthermore, the vast majority of eukaryotic DNA is what we call intronic. In other words, it is eukaryotic DNA that is intervening sequences that don't end up making it all the way onto a messenger RNA. Can you guys see this picture? Everybody look at this. You see here it says exon, intron, exon, intron, exon. Yeah? See that? Okay. So the exons and introns, which gets kept in, some of these are in the messenger RNA that's going to leave, and some are not. Which ones go into the messenger RNA when it's done? Exons or introns? Exons get to exit the nucleus, yeah? Introns have to stay in the nucleus. Does that make sense? So the technical term is that they're intervening sequences and expressed sequences, but you could say that introns stay in and exons get to exit. I think that's a pretty useful, right? You guys get that, though? So a lot of the DNA in you is not ultimately turned into a messenger RNA. It never makes it that far. It gets spliced out, and then those pieces get disintegrated, and the remaining exons get glued together. You guys got that? So here it is, the same picture, only this is from your textbook. I got this picture from another textbook. The colors are a little better. I just like it a little better because we use pink instead of green, and I don't know why I like that better. But it's basically showing the same thing, yeah? This is showing the level of detail I expect you to know. I don't expect you to know about this methyl cap and poly A tail. It's shown here. Don't worry about it, okay? All right. This, though, right, you should understand all these different things here, yeah? I think this picture, I would put a little star around this, yeah? Put a little circle around it. You should understand what's going on in this picture, okay? What's going on here? We've got DNA separated, messenger RNA coming through. What's that? That's transcription, yeah? Then what's going on here is introns are being removed, exons are being glued together, yeah? Once those introns are spliced out, the messenger RNA goes out to this thing. What's this brown thing? A ribosome. And then these green barrel-y things with purple things on them, what are those? Those are tRNAs, yeah? And what's going on here? This is where tRNAs are getting all these different purple amino acids linked onto them, yeah? And we saw that in that other picture, yeah? Can you relate this picture to the other pictures we've seen, yeah? Okay. All right, so this picture encapsulates most of the salient differences between eukaryotic and prokaryotic gene expression, yeah? If we look at prokaryotic gene expression, we can see RNA polymerase makes a messenger RNA. The messenger RNA is shown in green here, and we can see that a bunch of ribosomes are gang tackling the same messenger RNA. Yeah? You see that? There's no mechanism, and the messenger RNA isn't even done being made. Yeah? Transcription isn't even finished, and translation has already started, not with one ribosome, but 2 or 3 or 4 or 5 ribosomes, all on the same messenger RNA that's getting clobbered before it's even completed. Yeah? This could never happen in a eukaryotic cell because the messenger RNA is made and significantly modified before a ribosome ever even sees it. Does that make sense? Okay. As far as the differences between eukaryotic and prokaryotic ribosomes, there's none in terms of the mechanisms. Other than that, because they're different, we can target eukaryotic ribosomes with antibiotics, or prokaryotic ribosomes with antibiotics. Differences between eukaryotic and prokaryotic ribosomes make targets for antibiotics, and we've talked about that already, yeah? Questions? All right, so should we answer this question? Which of the following is mismatched? Well, first of all, ignore C and D, right? I haven't talked about those, yeah? I should strike those out. It's either A or B. Which one is wrong? People are saying B is wrong. Is that the right answer? A is true and B is not, yeah? Why is B not correct? Yeah. Does RNA polymerase make a molecule of RNA? Yes, it does. It makes a molecule of RNA, but what does it make it out of? It makes it from a DNA template. Does that make sense? You guys hopefully have a picture in your mind that shows that. Yeah? Questions? All right, you guys want to take lunch? I've now got 10 after 12, yeah? Come back here at 12:40, and that'll be lunch, and then we'll do lab, and then class will end.